Well, it's a real pleasure to introduce Rayna Bell for her second talk. This is the last seminar of our joint IB-MBZ search, last but not least. And we heard a wonderful talk yesterday all about color in frogs and perception of color in frogs. And today I think we're going to hear about the other side of Rayna's research program. So I'll just remind you, she did her undergraduate work at Berkeley. She then went to Cornell, where she did her PhD with Kelly Zamudio, and then was back here as a postdoctoral fellow, and has been at the Smithsonian for the last 2 years, where she's the curator of amphibians and reptiles there. So Rayna is interested in processes guiding diversification and understanding the details of those processes as well as the patterns. And she's done a lot of work on reed frogs and work on island biogeography, and I suspect that's what we're going to hear about today. So please join me in welcoming Rayna again for her second talk.
So thanks everyone for coming. I'm just going to warn you that the first couple of minutes of my talk are me taking an opportunity to acknowledge the MBZ in some ways that it's played a role in my academic development over the last couple of years, because I never really got an opportunity to do that. So contrary to what these photos would suggest, I did not grow up loving amphibians and reptiles. And a real life-changing moment for me was randomly enrolling in herpetology as an undergrad here and taking herpetology from Jim McGuire. And it was in one of his lectures on amphibian diversity where he mentioned that over 100 new species of frogs were being described every year, and that this had been true for over a decade at this point, and that not only were we discovering lots of new species, but that we still knew very little about the basic biology of these new species. And that was one of the first times as a student that I realized how much we still had to learn about our world, even about these really big, charismatic, and obvious animals, let alone the smaller ones. And I realized that I would actually be able to make a contribution to this potentially, and so that really set me on this career path. So thank you, Jim. And alongside that, I started doing research in the MBZ in Craig Lawrence's lab, and I started working on amphibians and reptiles in the Australia wet tropics. And so getting experience with phylogeography in the lab and going into the field a bit through the herpetology course, and then eventually doing some fieldwork in Australia really cemented my interests in evolutionary genetics and field biology as being a core part of my research program. Another thing that I learned as an undergraduate here is that the MBZ is a great place to find collaborators, even when you move on to the next stage in your career. So I did my PhD at Cornell and left the MBZ behind physically, but in a lot of ways the MBZ came with me. So Brian Stewart was a postdoc here when I was an undergrad, and he was instrumental in helping me launch a field research program in Central Africa, and he came into the field with me 4 times during my graduate research, and I would not have been able to get started there and learn how to prepare voucher specimens had it not been for Brian. So he's been a major supporter of mine. 2 graduate students that were here when I was an undergrad continue to play an important role in my research, and I'll be talking a little bit about a project that Adam O'Shea and Matt Fujita and I have in common. And then Dan Portick, who we didn't overlap here when I was an undergrad, but we sort of led parallel graduate school trajectories where we were both interested in African frogs. And so we collaborated a bit when I was at Cornell, and then when I came here for my postdoc, we really solidified our African frog dream team, and we have a bunch of projects in common together now. And when I came back, I was reminded of how awesome MBZ undergrads are. So of course I'm a little biased because I was an undergrad here, but the next generation just as awesome. So these are 3 students that I had the opportunity to work with when I was here, and they were incredibly hardworking, asked really good questions, and have follow through on projects, and so I'm preparing, either have published or preparing manuscripts with these students, and that's been a really wonderful and unique experience, I think, just in terms of the quality of students that come through this program. So I'm now at the National Museum of Natural History, and these are the three aspects of my job as a curator that I take most seriously. So the first is continuing this basic biological exploration in Central Africa. I started that field research program as a PhD student, and it continues to be an important part of my work, so voucher-based biodiversity surveys. As I'm sure those of you who are curators who have spent a lot of time in collections know, there's an endless supply of collection improvement projects lurking places. So in the middle here I have one of these pictured. These are specimens that are part of the Huyton collection. So for those of you who don't know, Dr. Huyton is an expert in East Coast plethodonid salamanders and had a very prolific collecting career, and over 180,000 specimens in our national collection were contributed by Dr. Huyton. And it turns out that about 6,000 or so of these specimens he never prepared. They were just frozen whole. And so these date back to the 50s and have extensive data associated with them. Many of them are collected from the sites that Dr. Huyton visited throughout his career for his life history studies. And so although we've seen some progress in getting DNA out of historical or formalin-fixed specimens, these have been frozen at -20 for the last 50 or so years. So the one main limitation is that this is actually what happens when you open a freezer. This is what the salamanders look like. So they're just stuck on little scraps of paper that have some notes associated with them. And so a major project in the last year has been getting some technicians to help us prepare this collection and make it available to researchers. And so a new postdoc in my lab is doing a project looking at changes in genetic diversity through time in some of these plethodonid populations, and he'll be using some of this material that has been locked in a freezer for the last however many years. And then a third project that I started is identifying benchmarks for future collections in the National Museum. So most of our focus is on North American diversity, especially because, as I mentioned, we have nearly 200,000 plethodonid salamanders. But there are a lot of areas where we can still improve, and one of these is having genetic samples available for a lot of these North American taxa. And so a project that we just started, we're calling the Type Locality Project, and we're revisiting type localities for North American vertebrate species, and we started just focusing on salamanders that were described from the state of Virginia. So we're revisiting these type localities and collecting topotypic vouchers, and also documenting whether or not we can still find these species at the original locations they were described from. And so we've involved undergraduate students in all aspects of this project, teaching them how to plan field expeditions, how to collect and prepare voucher specimens, and then how to catalog and curate these vouchers in the national collection. And Mike, who's a grad student in ESPM, was visiting the museum over the summer, and he's actually in this photo. We're revisiting the type locality of Plethodon dixie, which is only known from these caverns in the state of Virginia. So that was a really cool one to get to see. Okay, so as Michael mentioned in my talk yesterday, I focused more on sexual dichromatism and vision evolution in frogs. And that's sort of one half of my research program, and the other half is focusing on diversification on Limbless Islands and on Oceanic Islands, and also in Central Africa, but I won't be talking so much about the continental work today. So what I am going to talk about is diversification in these three contexts. So the first is diversification on land bridge islands, then talking about how organisms colonize oceanic islands, and finally what happens once they reach those islands, how do they diversify in situ. So most people are familiar with evolutionary processes that are happening on oceanic islands. So where do we get new endemic species? Well, they can either disperse from the continent or from adjacent islands, and then diverge to become endemic to that particular island, or we can have in situ diversification of resident species on an island. And the contributions of these two sources of endemic species are related to different attributes of an archipelago and individual islands in that archipelago. So as islands increase in age, size, and remoteness, the contribution of in-situ diversification becomes more important, whereas dispersal tends to dominate in islands that are smaller or closer to continental sources and young. And there are lots of studies that look at these processes in a lot of archipelagos across the world, so notably the Hawaiian Islands and the Galapagos. So in contrast to oceanic islands, most populations on land bridge islands are recently isolated from the adjacent mainland. And that's because these land bridge islands are part of the continental shelf, and the seas are rising and retreating and either connecting or isolating these more upland areas on the continental shelf from the rest of the continent. And so we can imagine that animals that are good dispersers or plants that are good dispersers won't necessarily feel the impacts of these marine incursions very strongly. Whereas animals that are poor dispersers, especially across saltwater barriers like amphibians, will respond strongly to these temporary marine incursions. So I've been studying these processes in an archipelago off the west central coast of Africa. And so these islands differ in their geologic history, so I'm just going to orient you a bit to the archipelago. So the first island in the chain is Bioko. The volcanoes that comprise this island are about 1 to 3 million years old, and throughout this history, they've been connected and isolated from the continental shelf just in response to global glacial cycles and sea levels. Next, we have Principe Island. This island is about 30 million years old. It and the rest of the islands arose from the seafloor. They haven't been connected to the continent. And Principe, because it's so old, has actually eroded quite a bit. It's less than a fourth the size it was initially. So when it first formed, it was much larger than it is today. The next island in the chain is Sao Tome Island. It's about 13 million years, and it still has more or less its original footprint. And then the final island is really tiny. You probably can't really see, but this is a runway. So it takes basically, you can land a plane there, but it takes basically the whole island to do it. And this island is about 5 million years old. And if you're looking at this island chain, you might be thinking, this doesn't really look like a hotspot because the ages aren't linear, and it's not. So it's what's called a suture, and it's not at the convergence of two plates. And I've talked to geologists about this to try to understand what's going on, and they don't really know, it turns out. So the ages are more or less known, and these things appear to have arisen in place, but the exact geologic processes that explain the formation of these islands and also the Cameroonian highlands, which are an extension of the same suture, are poorly understood. And then one final thing that I'll note is that they increase in distance from the edge of the present-day continent. So these differences in their biogeographic history, so the size, the age, and the remoteness, and then the geologic origin of these islands is pretty easy to see when you look at the composition of endemic diversity on the islands. So Bioko Island has very high diversity, but very little of this diversity is endemic. It's mostly shared with the adjacent continent, and that's true across taxonomic groups. And by contrast, when we look at the oceanic islands, we have less diversity overall, but almost all of that diversity is endemic. And so there are a number of groups that have sister species among different islands in the chain. And there's species or groups that have diversified within islands in the archipelago. And so it looks like across taxonomic groups there's been a strong influence of dispersal from the continent to the islands, as well as diversification within the archipelago, resulting in this really high endemism. And for those of you who are wondering what this thing is, this is a limbless skink on the right. And so there are also a lot of endemics on these islands that are things that you don't really expect to see on an oceanic island. So things that we think of as being poor dispersers and that should have had a hard time colonizing an island by overseas dispersal. So I mostly focus on frog biodiversity, and frog diversity really nicely illustrates this overall pattern of diversity and endemism in the archipelago. So if we look at Bioko Island, this is just a sampling of the diversity that we have here, and almost all of these species are shared with the continent. If we look at Principe Island, we have 3 endemics. These are from 3 different families. So in this case, we would say 3 different dispersal events from the continent. On Sao Tome Island, we have 5 different amphibian species from 4 families, and this is a Sicilian, so again, another animal that you don't expect to find on an oceanic island. And then Annobon has no amphibians, so I will not be talking about it for the rest of today. And I haven't been there yet, but I do want to go just to complete my visual observations of the archipelago. There are endemic herps there, so there's endemic lizards and endemic snake, and lots of birds and other plants. Okay, so I initially chose this study system and frogs because it seemed to me like a really good region to be looking at the evolutionary processes that are happening at these different timescales and also in these different biogeographic contexts with a shared fauna. So looking at frog diversification in these different evolutionary contexts. So if we look at patterns of divergence between Bioko populations and their counterparts on the continent, and a few studies have done this using a combination of genetic and sometimes morphological data, the common pattern seems to be that for the most part, Bioko populations are genetically distinct, but it's very shallow divergence and is on par with what we'd expect just between different populations on the continent. So not necessarily strong evidence of the marine barrier being important, or maybe just being very recent and there not being much time for divergence to accumulate. But in some instances, we find really deep divergence between Bioko populations and the rest of the continent, and this can be accompanied by morphological divergence. So this is an example of a gecko that Adam Lachey and Matt Fujita have been studying for some time, and they've accumulated a very large data set and have demonstrated that the Bioko population of the species complex is a unique species, and they described it recently. So we're seeing these 2 different types of responses to this shared history of marine incursions, isolating and connecting Bioko Island. And so in thinking about how this can happen, I think it's useful to think about just the baseline geologic processes that are happening, or in this case, the climatological processes that are happening that are influencing this history of vicariance and connectivity. So if we are looking at the most recent period preceding this one, where sea levels were high, we can see that populations on Bioko would have been isolated from their mainland counterparts, and if these organisms are poor dispersers across salt water, that we begin to accumulate genetic differences between these populations. Then when sea levels retreat and the land bridge is exposed, these populations can come into contact for a period of time and then become isolated again, like they are presently, and that the gene flow would cease, and that perhaps some of that initial genetic divergence that had accumulated during the period of vicariance has now been obscured by this period of admixture. There might also be a scenario where some organisms are more recent arrivers to this land bridge island, and that they would have dispersed overseas to get there. So perhaps colonists disperse overseas to reach the island in a more traditional founder event colonization, then that this population would grow over time, and then subsequently follow the standard connectivity and isolation pattern that we see. So sea levels retreat, we have a discrete admixture event, and then vicariance secondarily when the sea levels rise again. And then finally, for some organisms, we might expect that dispersal has been an important part of their history across the species range throughout. So perhaps not complete admixture, but infrequent, persistent episodes of dispersal just by chance crossing this very narrow marine barrier. And that, again, we would expect that admixture is greater when the land bridge is exposed, but that there isn't a complete cessation of gene flow when the sea levels rise again. So if you're thinking that this sounds like a lot of very complicated potential evolutionary scenarios, I'm with you. And And it's been challenging to try to think about this system over the years. So one of the first things that we did was just model the connectivity of the land bridge island to the continent, and we can only really do that for a relatively recent time period, so going back 120,000 years ago, and modeling both changes in sea level, and so when is the land bridge exposed, and then for a handful of species, generating niche models of the climatological variables that explain where we find these species today, and projecting those through time to understand whether the exposed land bridge would have been suitable for that species to disperse to the island. So we generate these niche models using occurrence data of where we find the species today throughout their range. Then we project these niche models back through time, again including these changes in sea level in our models. And then we estimate connectivity, so these black boundaries delineate the present day edges of continental Africa and Bioko Island, and green colors are high connectivity or high suitability for a species. So we can make an estimate of connectivity between Bioko and the rest of the species range during these periods when the land bridge is exposed. So we did this for 4 species, and here we're going to be going back in time from the present, and then this estimate of connectivity is basically not just when is the land bridge exposed, but also how suitable is the climate across the land bridge for a given species. And we included 2 reed frog species that are present on Bioko and 2 that are absent. So what we see is for the 2 species that are present on Bioko, we have this window of time where there's high connectivity across the land bridge in the last 120,000 years. So this gives us an approximate window of when we think one of these discrete admixture events could have happened. And then, surprisingly, for the 2 species that are absent from Bioko, we actually don't discover very high connectivity for them across the continental shelf. And so it's possible that this helps explain why we find some, but not all of the continental diversity on Bioko, even though it is very close to a biodiversity hotspot for amphibians. So if we look at patterns of mitochondrial divergence in a handful of these reed frog species, and here we have the Bioko populations in pink and Cameroon in orange. For this reed frog, the forest reed frog, what we see is mostly distinct haplotypes on Bioko, but with one individual in Cameroon clustering with these. So this might indicate some kind of incomplete lineage sorting or perhaps recent dispersal between these populations. And if we look at the period of divergence, the confidence intervals overlap with this period of high connectivity that we have from the niche modeling. And these types of dating analyses are always going to overestimate dates just based on how these methods are working. So I think it roughly coincides with this period, but certainly it looks like it's a more recent event. These things haven't been isolated for all that long out there. If we look at a second reed frog species, again, this is one of the ones that was included in the niche modeling. We see 2 distinct mitochondrial groups and a Cameroon population nested within, and the ages of these groups are a little bit different, but again, roughly coinciding with this period of recent connectivity. And so this pattern might indicate dispersal from Bioko back to the continent at some point, or it could just indicate multiple source populations, so that different parts of the Hyperoleus ocellatus range colonize Bioko across the land shelf of the land bridge, because it is a very broad geographic area that was contiguous. And then finally, we collected data for this 3rd species. This is a leaf-folding frog. It has a pretty similar ecology to these reed frogs. And again, we're getting a distinct haplotype group on Bioko Island and roughly the same divergence estimates for this species. So just looking at these patterns, we definitely cannot differentiate among these very confusing scenarios that I showed you. And so what we did was take a demographic modeling approach with Genome-Wide Snip data to try to address this question a little bit more convincingly. So in this approach, we develop a series of models, and this is work that Dan Portick was instrumental in. He's a collaborator on this project. And so we have an initial bifurcation in the models where a scenario that is more consistent with vicariance versus a scenario that's more consistent with a founder event. And so the main difference is that the founder event includes a change in population size, whereas the vicariance one does not. And then within each of these models, we can add these different confusing components of evolutionary history. So we can add a history of infrequent dispersal throughout the history of this population, and we can also add these discrete admixture events. So we have a whole series of models that we can test with our data. So first we collect this genetic data set, and Kristen Charles, who's one of the awesome MPZ undergrads that I mentioned, collected this data set. And so we have data from throughout the species range on the continent and also on Bioko Island. Then we look at the genetic structure of this species so that we can identify the continental population that's most relevant for our demographic modeling. And then we compare all these demographic models and use the joint site frequency spectrum to test which of these models is the best fit to the genetic data that we observe. So what we found was that a model of vicariance with a late discrete admixture period is the best fit for the leaf-folding frog. And for those of you who are more visual, this is the cartoon version of what we think this looks like. So vicariance when the sea levels are high, followed by discrete admixture when the sea levels retreat, followed by vicariance when the sea levels rise again. We also wanted to look at whether or not this divergence corresponded with any morphological divergence, like we've seen in a few other herp species. So these leaf-folding frogs vary in dorsal color pattern. They can be roughly classified into these two groups, type 1 and type 2, and there's a lot of variation throughout the species range on the continent. And so we thought maybe this variation corresponds to something meaningful, but it turns out no, it's just a polymorphism. And so we see both of these dorsal colorations in the Bioko population, in the North population, which is the most closely related population, and in the other populations in our sampling. We also looked at some basic morphological variation, and this teal group is a population that's been described previously as a distinct subspecies, so we do see some morphological divergence in that subspecies, and we also found genetic divergence in that group. But otherwise, for the rest of the species range, including Bioko, we didn't find any evidence of morphological divergence in these frogs. So the next steps, we've collected genetic data for the other two reed frogs, and we'll be putting them through the same pipeline of demographic models to see whether they're responding in similar ways as the leaf-folding frog is to this history. And we also have data for a bunch of different kinds of frogs. And this is part of Matt and Adam's big West Africa Comparative Biogeography project, where we have a bunch of African amphibian scientists coming together and pooling all our samples to be able to ask these really interesting questions about diversification and the biogeographic history of species across the continent. And so for this Bioko comparison, we're focusing on these 7 species, and these represent a diversity of body sizes and reproductive modes and behaviors, and so we're really interested to see whether we find a common pattern across these groups or whether they all differ in their own ways in response to this shared history. Okay, so now we're going to move to the Oceanic islands and the archipelago, and in this case we're not going to be testing different models of how things would have gotten there because they had to get there by overseas dispersal. But how did they get there? So, as I mentioned, there are several endemic amphibians from different families that are present on these islands, but these aren't the only enigmatic things that you find there. So, Principe and Sao Tome each have an endemic species of shrew, and shrews are not things, again, that you would expect to find on an oceanic island. They have to eat every couple hours. They're probably not very good at swimming in salt water, so it's surprising that these things have made it out there multiple times. We also have several phosphorial reptiles, so I showed you that limbless skink, but there are also these little blind snakes, and these, again, are not things that you expect to find on oceanic islands. And there's also the caecilium that I mentioned. So the prevailing hypothesis for how these things would have gotten out there are vegetation rafts that would be swept down these large river drainages into the Gulf of Guinea. And there are 3 main rivers that reach the Gulf of Guinea that are hypothesized sources for these types of rafts. So the first is the Congo River, which reaches the southernmost part of the Gulf. And the Congo River is a huge river that spans most of Central Africa, and it's the primary drainage for most of the Central African forests, so it has a huge discharge. The second river is the Niger. This is the major drainage for all of West African forests, and it reaches the northern part of the Gulf. And the third is the Ogooué River, which drains most of Gabon's rainforest, so the western part of the Central African rainforest. So in case you're having a hard time picturing what these vegetation rafts would look like, this is a gratuitous artist rendition of what it would be. I don't know that they really would have been this big. These are all trees. But the idea is that you basically have a mini ecosystem getting shunted out to sea and being swept down from these river drainages. So although the mouth of the Ogooué River is the closest to the islands, the currents in this part of the Gulf direct freshwater plumes from the mouths of these other rivers towards the islands. And so it's conceivable that rafts from these rivers would be making it to the islands. And in addition to that, the surface salinity on these freshwater plumes is much lower than typical seawater. So as crazy as this mechanism sounds, it's certainly conceivable that it's happened over the last 30 million years a few times, because these islands have been out here for so long. An alternative hypothesis is that organisms have dispersed down the island chain following a stepping stone pattern. And this is certainly possible in that we have a lot of shared sister taxa, particularly between the Oceanic islands, less so with Bioko Island. But that is another possibility, is that we have stepping stone colonization. So we can test these different hypotheses by looking for the most closely related species on the continent to these island endemics, and reconstructing phylogenetic relationships and identifying where these potential source populations would have come from. And so a few studies of these groups have included the island endemics plus representative specimens from the same genus shared on the continent. And this emerging pattern is that a lot of the island endemics share a closest relative in East Africa, which seems crazy, but again, the Congo River does initiate out in East Africa and drains across the whole continent. One major limitation of these studies has been the availability of samples from West and Central Africa to include in these phylogenies. And so that's where I've really focused my efforts, is trying to fill these sampling gaps, because for obvious reasons, they're limiting our interpretation of these patterns. So I chose to focus on the reed frogs because they're the only amphibian group that has diversified within and between islands in the archipelago, and so I thought this would be a nice group to focus on. When I first started working on them, this mitochondrial phylogeny had come out that identified a species complex on the continent that was closely related to the island endemics. And so that was very helpful in narrowing down my initial search. Reed frogs are a very diverse group of over 150 described species. And so initially I thought I was going to have to resolve the entire phylogeny of the group to be able to identify the closest mainland species. But luckily, I was able to do this project without doing that, because Dan and I are just finishing that project now. And yeah, it's been a long one. So the distribution of the Sinomami of interest complex across Central Africa encompasses both the Ogooué River and the Congo River, but not the Niger. So we can eliminate that as a potential source just based on the geographic distribution of Sinomami of interest. And then for the stepping stone colonization hypothesis, Sinomami of interest is one of those species that we included in the niche modeling, and it does not occur on Bioko. So it's unlikely that these frogs colonize the Oceanic islands by first reaching Bioko and then dispersing down the chain. So I took a phylogeographic approach with this group, in part because it was a species complex, but also because of its range. We wanted to be able to narrow in on the populations that are most closely related to the island endemics. So here we have sampling throughout the Sinomami-Ventris range, and you can see that the green populations surrounding the Ogooué River, or perhaps the westernmost part of the Congo River, are the populations that are most closely related to the island endemics. And so what this suggests is that populations from West Central Africa are the ones that initially dispersed and colonized the islands, and it's in contrast to this East Africa pattern that we kept seeing. So that was a mitochondrial biogeography, and I followed up that study with some rad-seq data and found the same relationships. And so again, what this pattern of divergence suggests is that we had dispersal somewhere in West Africa, first to Sao Tome, based on the pattern of divergence among the species. Diversification in situ on Sao Tome, resulting in Hyperoleus thamensis on the right and Hyperoleus molleri, and then dispersal from Sao Tome to Principe, resulting in a third species on Principe Island. And if you're looking at the divergence times of these events, these are all relatively recent in the geologic history of these islands. So Sao Tome, like I said, is about 13 million years old, and based on our estimates, these frogs only arrived there in the last couple million years. So these islands are still accumulating endemic diversity. So now that I had roughly resolved the reed frog history, I wanted to move on to one of the other groups for which we had no real information about what their most closely related sister species is on the continent, and that is the Principe giant reed frog, or tree frog, sorry, that's this guy. This is one of the largest tree frog species in the world, and based on some shared morphological traits and a limited mitochondrial data set, it's hypothesized to be closely related to this group of other large-bodied tree frogs in the genus. And these three species distributions are shown in these three different colors, and as you can see, they encompass all three potential river systems that could be a source of these rafts. And one of the species also occurs on Bioko Island, and Principe is the next island in the chain, so this stepping stone pattern is a possibility for this species. So I had an REU student last summer who helped with this project, and I thought it would be pretty straightforward because, again, we had a hypothesis of who we thought would be most closely related. And unfortunately for Kyle, the main thing that we could say at the end of his summer internship was that Palmatus, the Principe species, is not closely related to any of these giant tree frogs, and we have no idea what it's closely related to. It's very, very low support for any of these relationships. So this is just based on a mitochondrial gene, and we don't have complete sampling of the genus, so we're definitely going to need to increase our sampling and get more robust markers. The one saving grace for Kyle's morale was when he realized that this meant gigantism had evolved multiple times in the genus, so that was a nice recovery moment. And when we look at the distributions of these different species that are potentially closely related to Palmatus, what we see is that they occur basically in every biogeographic region of Africa, just reinforcing that we're really going to need to resolve the phylogenetic relationships in this genus to answer this question. And one possibility is that, Principe is 30 million years old, and it's possible that Palmatus has been out there for a very long part of this history. So it's possible that the original sister species on the continent have gone extinct, or that they've just been divergent or isolated for so long that we're going to have a really hard time resolving where this species fits into the rest of the phylogeny. So we'll see what happens. Okay, so now I'm going to shift to in-situ diversification within the archipelago. So to remind you of the players, we have Hyperoleus thamensis, which is the giant reed frog, the sort of bombina-looking belly frog on the right there, and then Hyperoleus molleri. So those are both endemic to Sao Tome. Hyperoleus thamensis occurs in closed canopy forest and breeds in tree hole cavities, and Hyperoleus molleri occurs in more open habitats and breeds in any standing water available to it. It's not picky at all. And then Hyperoleus drusei is very ecologically similar to molleri. It's just restricted to Principe Island. And so looking at this mini radiation of frogs, I was interested in the in-situ diversification on Sao Tome, and then also the history of overseas dispersal between Sao Tome and Principe. So, and I don't think that's how it happened. I really like that cartoon. Okay, so perhaps unsurprisingly, when we look at genetic variation between molari and drusei, we see that they form two completely genetically distinct groups. So what this indicates is that dispersal happened probably once in the history of these species, but it's not ongoing. And at the time of this study, this was considered one species, but based on these genetic data and also examining literally hundreds and hundreds of specimens, I ended up describing the Principe population as a distinct species. So then focusing on diversification on Sao Tome Island. So I mentioned already some ecological differences between these species in terms of where they're found and where they breed. And the Hybrales thamensis is called a giant reed frog because it's huge. It's about this big. And molari is a regular sized reed frog. So they differ in body size. And they also differ in ventral coloration. And what I started noticing in the field was that I was finding individuals that were phenotypically intermediate. So they were intermediate in body size and also intermediate in ventral coloration. And so when I collected genetic data for these individuals, I would often find mismatched mitochondrial haplotypes with nuclear genetic assignment probabilities, and a lot of individuals that had mixed assignment probabilities. And so I wanted to know whether or not these things were hybrids. And I used this new hybrids approach, which classifies individuals based on genetic data into different categories of hybrids. So F1, F2, and backcross hybrids. And this kind of looks like a structure plot, but each vertical column is an individual, and the color represents the assignment probability to one of these different hybrid classes. So these all have very strong assignment probabilities to specific classes. And so what we can see is that we have these three sites where we have a really high abundance of hybrids. More than half of the individuals that I find at these sites are hybrids. And that it quickly tapers off when we leave these sites. And so if we look at a cline, so here we're looking at a mitochondrial cline and then a combined nuclear cline, it's consistent again with this really strong tapering off of the hybrid zone. It's really just restricted to a couple sites. And the same is true when we look at phenotypic data. So here we're looking at ventral coloration and body size. So what's happening at these sites? Well, this is what these sites look like. So they tend to be these agricultural fields right at the boundary of the protected forest habitat on this island. And when I find frogs at these sites, I find them in artificial breeding environments. So either in tarps that are full of water or barrels that people are using to water their crops. And so what I think is happening is that these artificial breeding sites are providing opportunities for these frogs to encounter each other where they normally wouldn't because of their differences in breeding site choice. And so if this is what's happening, then we would expect that this contact between these species is relatively recent. Either that or that there's really strong selection against hybridization, and that's why we don't see hybrids outside of the hybrid zone. But given that we're seeing multi-generation hybrids, they appear to be viable and able to reproduce. So we don't have very strong evidence for there being very strong selection against hybridization just based on the viability of the offspring. So to get a better handle on what's going on here, I've been busy collecting samples from all different parts of the island. So I haven't collected genetic data from all of these sites yet, but I have samples from 2 additional contact zones between primary forest and more open or disturbed habitats. So we'll be able to look at parallel hybrid zones across these transitions. And what's very cool about these transition zones is that Drosophila appear to be hybridizing in the same place. So a lot of Drosophila biologists know about Sao Tome, and it's because there's an endemic Drosophila that's been really well studied from there. It's Drosophila santomea. And it's also a more primary forest species, and it hybridizes with a more disturbed habitat species also at the interface of these 2 habitat types. So we have parallel hybrid zones in frogs and in fruit flies, and possibly birds as well. And so the groups that are studying these different organisms and I have been being strategic about our field sampling so that we overlap, and we're also trying to be strategic in our methods. So we're looking at genetic divergence and also phenotypic divergence across these contact zones. And we're hoping that by looking across these taxonomic groups and patterns of introgression, we'll be able to get at how old these contact zones are and whether we have replicate suture zones across the island at these points of contact between primary forest and more disturbed habitats. Okay, so finally now I'm going to talk a bit about the species recognition traits that are involved in divergence in this system. And I'm going to be focusing on advertisement call. So for those of you who don't know, frogs produce calls to attract their mates, and females usually respond to calls from males of their own species. And this is considered to be one of the most important prezygotic barriers in frogs. So the main elements of call that can vary between species and that females pay attention to are the frequency of the note and also the number of pulses that are repeated. And so, although these are diagnostic, sometimes mistakes happen. So this is a parent amplexus from Principe Island, where there are only 3 species of amphibian, and where mistakes shouldn't happen very often. And these are frogs from 2 completely different families. So it's not a perfect system, but it usually works pretty well. And in the case of the Sao Tome hybrid zone, we would expect that it's not a perfect system either because we're finding lots of hybrids in this contact zone. So one of the main questions underlying this work was how divergent the calls of the 2 species that are hybridizing are, and what kinds of calls do the hybrids make. So here we're looking at body size and dominant frequency for 4 species. So we have the 2 species that are hybridizing, the most closely related species from the continent, and then the species that's endemic to Principe Island. And what you can see is there's a strong correlation between body size and the dominant frequency of the call the frog produces, regardless of evolutionary history or relationships among these species. It's just a very strong correlation. And that's true for basically all frogs that people have looked at so far. They're just really tightly linked traits. And when we look at hybrid males, we find that they have intermediate body sizes, and correspondingly they produce calls with intermediate dominant frequencies. So now that we've quantified some of this variation, we can go back and try to get a handle on how these calls might be diverging, whether there's any potential for reinforcement in this system, and whether females are discriminating against hybrid male calls. And then this was a somewhat unexpected result. So here we're now looking at average pulses per call instead of body size. And what you can see is, for some reason, the frogs on Principe produce so many pulses. And in other systems, this has been shown to be driven by female preference. And so, again, we're interested in looking at whether there's a female preference or discrimination for calls with different pulse numbers, and whether there's a preference for calls with more pulses. So that's where we're heading next. Okay, and so with that, I'll just say that I've been working in this system for the last 10 years, and it's been a privilege to work in these different countries and with this team of people. And I'm really excited about the potential to keep growing this research program here. It's definitely a biodiversity and natural history inspired line of work, and the Museum of Vertebrate Zoology has a really strong reputation for supporting that kind of research, and it would be a privilege to continue in that tradition. So I'll thank my many collaborators and take any questions.
Okay, if somebody could get the lights, we have time for questions.
Are the frogs in San Tome separated by elevation, or is it habitat here?
So initially I thought it was elevation based on my first transect that I had data for. And now that I've collected specimens from other parts of the range, I was surprised to find the giant species down at about 200 meters. But you do have primary forest down at 200 meters. And I also wanted to test, so often things, when you see patterns that are associated with elevation, they're associated with environmental variables like temperature and precipitation. And so I did look at those, and if you look at how those vary across the island, there is a pretty strong gradient even though these islands are very small. And the major transition between those environments happens roughly with where the hybrid zone is. So I did an analysis to look at SNPs that are correlated with those environmental variables, and we do find a few that are. And when we look at the declines of those SNPs, they're very gradual. So they're much more gradual than we see in the overall genetic background for the nuclear data, the mitochondrial genes, and in the phenotypes. So there are some things that I think are correlated with elevation and these environmental variables, but I don't think they explain where we find the species or the hybridization that we're seeing. Yeah.
Could you talk a little bit about how you arrived at the conclusion that Molari, I think it's on Principe, is derived from the Sao Tome and not the other way around?
Yeah, so it's based on the pattern of divergence among the species, but also patterns of genetic diversity. So the Principe population is even more genetically depauperate than the Sao Tome lineage, which is consistent with sequential bottlenecks. So there's basically one mitochondrial haplotype on Principe and very, very low heterozygosity. And in general, these species have very low heterozygosity, but it's especially pronounced in Principe. Yeah?
How big were the islands a few million years ago, and some of these events may have taken place? You mentioned Principe is highly eroded, and yet San Tome is significantly larger. So Principe must have been larger once upon a time?
It was, yeah. It was probably about 1,000 square kilometers. So large is relative.
And would that affect the results much, do you think?
You mean in terms of the demographic history of the species? Potentially, but it's been, I don't think that a million years ago Principe was 1,000 square kilometers, if that's what your question is. Yeah.
Do you have, for your modeling of connectivity over the last 120,000 years, do you have direct geological constraints on local relative sea level, or are you sort of assuming that the topography has been static and just using a global sea level reconstruction?
Yeah, it's global sea level, and in the last 100,000 years, I don't think the geology of the continental shelf has changed all that much from present day, but.
Shouldn't you, I don't know, some of these volcanic regions, there can be significant rise and fall of base level that might add some noise.
Yeah, we didn't include that.
Yeah, it might not be possible to constrain one.
Yeah.
So are there storms or hurricanes or anything that also might be contributing to animals flying around between these islands? Or
There are storms. Yeah, I think it's possible that things, especially things that can fly, are moving between the islands in different ways than things that would be rafting. So for the birds, for instance, we don't see the same pattern of divergence. There are endemic species, but it's not necessarily this south to north colonization like we see in a lot of the things that we think would be rafting, so the amphibians and the reptiles. Yeah.
Yeah, curious if you had any information on how quickly the gigantism evolved convergently in some of the tree frogs, and then if there's any ecological correlates that might suggest why that was selected for.
Yeah, that is another reason that we need a phylogeny for the group. So there's a lot of variation in body size in Leptopilus tree frogs, and we don't know why they get big. And we also don't know how long the Principe species has been on Principe. So it certainly fits the island giant paradigm.
I wonder
but
if there's any evidence that gigantism doesn't last long, like if extinction rates are higher. I mean, if they're really young, maybe it's just not maybe there's a problem with it, or I don't know if it lasts. I'd be curious, are there any old lineages that are giants within the tree frog group?
we don't know because we don't know how they're related to each other. So you can't even really make an inference about gigantism until you know what the closest relatives are and whether there's actually shifts in evolutionary rates of body size. So yeah, there are definitely limitations to trying to study these groups that we know very little about, but we're getting there. And yeah, that would be a great thing to ask.
Rayna, what's the model for speciation between these two? So you mentioned that you see a similar pattern in Drosophila with São Tomé and Yacuba, and then you mentioned the birds as well. So, I mean, that if you see similar patterns like that, and you suggested that this was secondary contact and that it was recent, is there a vicariant?
How did they diverge in the
Yeah,
first
was there an
place?
allopatric
Yeah,
vicariant event?
we
or what?
don't know. So it's a pretty small geographic area. And in the case of the Drosophila hybrid zone, we actually think Yacuba is a recent arrival associated with human colonization. So that hybrid zone they didn't diverge on the island, basically, as far as we know right now. With the birds, it's very contentious because, of course, in-situ diversification for birds on islands isn't supposed to happen. And they have a much more complicated scenario with other species in this group on Principe and a little islet off of Principe. And so that history might involve divergence in allopatry between the islands and then recolonization from Principe to São Tomé for the birds. And that's a possibility for the reed frogs as well. Again, I'm basing the colonization order in part on the genetic diversity that we see, so the fact that it's so low on Principe compared to São Tomé. And another thing that's a little bit odd in this case is the divergence in breeding habitat between the species. So not just the habitat, but also that Fomensis breeds in tree hole cavities, which happens in frogs, but it's a very specialized reproductive mode, and we don't fully understand the drivers of why you would want to breed in tree hole cavities. There are a lot of disadvantages to doing that. And we also don't understand why they're different in body size, because in terms of the habitats that we find them in, they're both arboreal. We don't know why this one has this crazy ventral coloration. So yeah, so it's not entirely clear. I think there was a period of allopatry, but I don't know what would have driven that initial allopatric divergence.
So on Sulawesi, we don't find, the frogs that we find at high elevation are direct developers. And these guys are egg layers, right? So I'm just curious if there's a lot of river habitat or stream habitats or whatever the habitat is that Molari is breeding in at high elevation, or if that might be the reason why these things are
That's what I
tree hole
thought
specialists.
initially, but then again, I've been finding Fomensis down at 250 meters elevation now on the southern part of the island where we have more intact closed canopy forest.
In Sympatria with Molari?
Yeah, right, again, at the edge of these agricultural fields and primary forest.
Do you find Molari in good habitat anywhere?
Not really.
Interesting. How about on Principe with Druzy?
Yeah, you can.
So you have DD-RAD data from hybrids as well?
Yeah.
So the phenotypes are really different.
Yeah.
And the hybrid area is pretty small. Can you do admixture mapping to try to get at the genes that underlie those phenotypic differences? Have you thought
I'm so
about
glad
that?
you asked. Yeah, we're sequencing genomes of the two species right now, and that's exactly one of the things that we'd like to do after. So we'd like to get a better estimate of when these things came back into contact, whether it really is recent or not, and then hopefully they are admixed enough, especially these multi-generation hybrids, that we'll be able to do some admixture mapping. Yeah.
You mentioned different factors impacting the biogeography of this system in the islands, but what about extinction? Do you have some information?
Yeah,
How would
I mean, we
it fit?
certainly think that extinction, just given the age of the islands, is playing an important role, and especially given that it looks like some of the things are recent arrivals. And so until we have a better picture on how long things have been there that are there presently, whether, like if all of our estimates are in the last 5 million years for everything, including Principe, that's going to be pretty suspicious, right, for a 30 million year old island. And so then I think we'll have an idea of the impact extinction is happening. It's always very hard to estimate the impact of extinction, but I think if we have really recent origins for everything, that would be one way of getting at that question. In terms of fossils and that sort of thing, I don't think there's any hope for frogs. Maybe for some of the other groups. But, yeah.
In the hybrids, were those phenotypes that were intermediate actually good predictors of genetic relatedness, or were there some that was it misleading in some cases? Like no spots, but
yeah, I place bets with myself when I catch a frog of whether it's going to be a hybrid or not, but I don't do it in a systematic way. And I haven't developed a hybrid index for the phenotypes yet to be able to go back and do that in a more realistic way. But so far, if I think something is some kind of hybrid, I'm usually right. But I'm kind of cheating because I'm in a place where I know I can find hybrids, and if I'm in that kind of habitat and something looks a little funny, then it's usually a hybrid. Yeah.
So I have a question about the daddy analyses that seem really cool. Do you get significance bias with those analyses? Like, do you have a lot of power in your data to distinguish these alternative demographic models, like effective population size changes through time that allow you to distinguish between these complicated?
You do. You can compare the AICs of the different models, but yeah, it depends. I mean, so this model is the one with the strongest support, but there are other models that have similar support and that differ in one or two of the parameters. But all the most strongly supported models are vicariance and not founder event.
You had a question in the back?
I was curious, are the natural predators for these frogs on the continent, evident on the islands?
So, as I mentioned in my talk yesterday, a big limitation in understanding diversification of these frogs has been that we don't really know that much about their predators. So one thing that is a little different on the islands is that at every single site that I find these, I'll find usually one brown house snake just hanging out. I've never seen one eating them, but there's one tree, for instance, where we find Hyperoleus thamensis, and it's just one tree hole cavity, and one of my collaborators, Bob Drews, has been visiting this tree for decades. And one time when I went there was a snake coiled right at the entrance to the tree hole cavity. I was like, no, you're going to decimate the whole species. So yeah, so there are snakes at every breeding site, basically. So I feel like they're probably there because they're eating the frogs, but I haven't seen that happen. We've seen spiders eating these frogs. I showed a nice photo of that yesterday. And then on the continent, we're also limited by predation data. So there's evidence that primates eat frogs, birds eat frogs, snakes eat frogs, obviously, and I see a lot of spider predation, but we don't have systematic data at all for any of these places, and so making that comparison isn't really possible yet. But I've certainly thought about, what's different on Sao Tome that this frog got big and colorful and is breeding in tree hole cavities, and very few of the reed frogs on the continent do either of those things, let alone all three. So I don't know if those traits are linked in some way, but I've been thinking about it. So.
Okay, if there are no more questions, let's thank Rayna once again.